Assalamu alaikum. So let's go into something really exciting. So let's talk about DNA cloning. Okay, let's define DNA cloning. So DNA cloning is basically a technique that we use to make pieces of many copies of a piece of DNA. And we use a certain biological system for that. Now, we can use bacteria mainly. We can use yeast as well because they're easy to manipulate, they are easy to handle. Or we can do DNA cloning on human cells in culture, in laboratories, that is. So, or we can use a cloned DNA to have bacterial cells or human cells express specific genes, like the insulin gene, for example, or a gene that produces an enzyme or whatever. So, and this is done by creating a recombinant DNA, which is basically a DNA that is a that is a that is made of different pieces of DNA from different sources. So we say human DNA, and we combine it with bacterial DNA. So that's a recombinant DNA. So basically, we have a piece of DNA that contains another piece of DNA from a different source. Now, we have one DNA that is called a vector. It's the carrier, and we have the gene of interest or the piece of DNA of interest. Now, usually it can be a gene that produces a protein, or it can be non-coding RNA, okay, that is not translated, whatever. And in order to do this, we use restriction endonucleases. So let's start talking about, let me remind you of restriction endonucleases and what they mean exactly. So restriction endonucleases are bacterial enzymes that cleave DNA. And what they do, basically, well, they recognize restriction sites, short DNA sequences, and they cleave within these sites, forming restriction fragments. Okay, and these are natural enzymes that exist in bacteria, and the natural purpose of these enzymes is to cleave bacteriophage DNA. So bacteriophages are viruses that can infect bacterial cells, and they take over these bacterial cells. They infect these bacterial cells. So they insert their own DNA, and then they take over. So what bacterial cells do is that they express restriction endonucleases that cleave the bacteriophage DNA so that they cannot control these bacterial cells. So humans identified these enzymes, and they said, how can I take advantage of these enzymes? So they use them for cloning. Now, what I did not tell you is the following. Actually, I told you that before. Basically, these restriction endonucleases uh, can cleave restriction sites that are palindromic, meaning that, well, they have palindromic sequences, meaning that you can read the restriction sites from either DNA strand the same exact way. So here we have GAATTC, and you read it the same exact way from the other complementary strand, GAATTC. Now this is the restriction site of ACAR1. Hindi 3, small 1, also recognize palindromic sequences as well. Okay, this is what I did not tell you before, and that is these restriction endonucleases can make blunt-ended restriction fragments or cohesive-ended DNA fragments, restriction fragments. What we mean by that is, with the blunt ends, is that the cut occurs right in the middle, in the center of the restriction site. So EcoR5 can recognize this sequence, GATATC. Now notice again, it's palindromic, right? GATATC. the cut occurs between T and A, right in the middle, okay? And note the DNA that the restriction fragments have blunt ends. That is, the cuts are straight, right in the center, okay? On the other hand, note here, echo R1 makes a staggered cut, meaning that the cut occurs not in the center, rather on the sides. So the cut occurs between the G and the A. The G and the A, note that are not in the center, not in the middle of the restriction site, rather on the sides, creating cohesive ends. Ends that have overhangs, right? Now they are called cohesive ends because these ends are complementary to each other. So you have TTAA, and here you have TTAA. So the AA here can hydrogen bond with the TT right here. The TT here can hydrogen bond with the AA right there. So these are known as cohesive ends. Okay, now I'll tell you later on that these hydrogen bonds are reversible, right? So the two pieces of DNA can separate from each other because the bonding is reversible. Now, in order to have the two strands connected to each other again, you need the formation of phosphodiester bonds between this fragment and that fragment, between the A and the G, the A here and the G there. Now, if we have two different pieces of DNA from different sources, and we have them cut by echo R1, we're going to have the formation of several restriction fragments, but all of them are going to have cohesive ends. So we can have the formation of hydrogen bonds between these two different fragments from different sources because they have ends that are complementary to each other. And then we can form, we can have the formation of phosphodiester bond between the A and the G, the A and the G right here. So we have what is known as a recombinant DNA. And a recombinant DNA is a DNA that is made from different sources. And we use restriction endonucleases to create these DNA molecules. So note that I have now one piece of DNA that is generated from different DNA sources. Now again, we can have the addition of a DNA ligase in the presence of ATP because it's ATP dependent, so that we have the linkage, the permanent, strong linkage of this phosphodiester bonds between the DNA fragments. Okay, so now we have what is known as DNA ligation. The enzyme is known as DNA ligase because it ligates, it connects molecules with each other. So what is DNA cloning? Well, DNA cloning means that you make copies of one thing. It's like a copy machine, okay? You have this piece of paper and you make 10 copies, 100 copies of that same piece of the paper. So what we do is that we can use bacteria. So these bacterial cells, okay, eventually you can grow them on dishes, and we have this bacterial cell forming a clone. Okay, so this is the bacterial clones or the different bacterial clones. Each clone comes from one single cell. So these all are identical to this, identical to each other, and identical to the original cell. Now, these cells right here are identical cells because they can come from one bacterial cell, so they are cloned. Okay, so if I say human cloning, it means that I'm making a copy of a person. So I have the same person copied over and over again. We have many copies of that same person, meaning that this person has identical DNA, identical genetic material. Before talking about how we clone DNA, we have to talk about DNA plasmids. These DNA plasmids are vectors, okay, vectors, meaning that they are carriers of the DNA that we want to clone, the DNA piece that we want to make um, many copies of. Now, these DNA plasmids can be cloning vectors or expression vectors. Okay, we'll talk about the difference between each. We'll talk about cloning vector. We'll talk about using plasmids as cloning vectors. Okay, so the idea here is what a plasmid is. It's extra chromosomal DNA in bacterial extra chromosomal DNA, meaning that if you look at a bacterial cell, we notice that each one of them has its own chromosome, the main genetic material that contains like E. coli, containing 4,000 different genes. So there's one copy of bacterial chromosome, or chromosomal DNA. But bacterial cells can also have plasmids. These are circular DNA pieces um, that can replicate independently. So each bacterial cell can have multiple copies of each plasmid, and these are small pieces of DNA. Now, naturally, plasmids contain genes. They carry genes that can give advantage to bacteria. For example, it can make them resistant to viruses. It can make them having antibiotic resistance ability. Okay, so that's what's important about is that they can replicate independently of the chromosomal DNA, and they can be engineered as well. Okay, that is, we can modify them, which so that they can do certain things. Okay, and when we say modify them, this is exactly what we're talking about. We're talking about genetic engineering. That is, we engineer genes um, or pieces of DNA, such as plasmids. Okay, so that's what plasmids are. Now, what we do is that we take these plasmid molecules and we make them function as vectors, meaning that we make them as carriers of pieces of DNA, target genes, for example, whatever piece of DNA that we want them to carry. And we create recombinant DNA, DNA made of different sources. So we take these plasmids, which are circular again, and we open them up with a restriction enzyme. We also take the target DNA and we add the same restriction endonuclease. So now both the plasmid DNA and the fragment DNA that we're interested in cloning have cohesive ends, and these cohesive ends are complementary to each other. Okay, so we open them up with a restriction nuclease, we combine them with each other, we just put them together in the same tube, and we add a DNA ligase. And what happens is that these ends, cohesive ends, are complementary to each other, so they can combine, they can hydrogen bond with each other. When we add a DNA ligase, the DNA ligase would form phosphodiester bonds between the two DNA pieces, and here we have a recombinant DNA, a plasmid DNA. Um, um, that is, the piece of DNA that we're interested in now is permanently part of the plasmid. That is what we mean with recombinant DNA technology. Recombinant DNA technology, the creation of recombinant DNA, the creation of DNA pieces that are made that come from different sources. So, in order to utilize plasmids as vectors, they have to have at least three features. One, they should have their own origin of replication. right? Remember that origin of replication. That is the starting point of DNA replication. So go back to the DNA replication lecture and read about the origin of replication. So they have their own origin of replication, at least one. All they need is just one. They don't need more than one, just one. And this is the starting point of DNA replication, and that what makes them replicate independently of the chromosomal DNA. They should also have a cloning site. Okay, that is a site where we can that is recognized by it's a restriction site, a site that is recognized by a restriction endonuclease. Now, the site is opened by this restriction endonuclease, creating cohesive ends, and so that when we add the DNA that we're interested in cloning, they can hydrogen bond with each other. And the third thing is that plasmids should have antibiotic resistance gene like ampicillin. Ampicillin is an antibiotic, and this gene makes bacteria resistant to ampicillin. Okay, that's exactly what we're talking about here. So that only bacteria that have this plasmid can survive ampicillin. They can, if we add ampicillin to this bacteria, they can survive. They cannot be killed. So these are the three things that are needed by plasmid DNA. A, origin of replication, so that they can replicate independently of the rest of bacteria. A cloning site, restriction site that can be cleaved by restriction endonucleases, so we can insert the DNA piece of interest, and the antibiotic resistance gene, so that only bacterial cells that contain the plasmid would survive if we add an antibiotic to them. So that's basically what we do. Okay, so we take the plasmid DNA, we take our insert, we add ECAR1 to each one of them, creating cohesive ends. Now these two ends are complementary to each other. When we add them to each other, they're gonna hydrogen bond. We add a DNA ligase, and the DNA ligase forms permanent linkage between the plasmid and the DNA of interest. Again, here we go. So we take DNA from human cells. We add ECOR1, let's say. Okay, again, we can add any restriction of the nuclease. Cohesive ends are created. We add the same endonuclease to the plasmid, creating cohesive ends as well. When we combine them to each other, okay, oh, sorry. So here we take the plasmid from bacteria, and we cut the plasmid with ECOR1. We combine them with each other, in addition to a ligase, and here we have the recombinant DNA. The recombinant DNA is then what we call transformed into bacteria. Transformed into bacteria. That is, we insert the plasmid into bacteria, and there are different ways of doing that. Now, the bacterial cell has a plasmid, has a recombinant DNA. Okay, now, again, same idea. Now, let's say we have bacteria cells that have a plasmid, and we have bacterial cells that do not have a plasmid. Now we add ampicillin to these bacteria. Now this bacterial cell would die because they are not resistant to ampicillin, and this bacterial cell would survive. Okay, because they have the plasmid that contains the recombinant DNA, the ampicillin resistance gene. Now we can multiply these bacteria, we can grow these bacteria, and let's say that we do the inoculation at 4 p.m. 4 in the afternoon. We let bacterial cells grow overnight. We're going to have millions of bacterial cells um, after like 16, 18 hours. Now, each bacterial cell would contain at least one plasmid, and these are the ones that would survive ampicillin. Now we're going to have billions of plasmid molecules, recombinant DNA molecules this way. So here we have a recombinant DNA, and we can clone our DNA of interest. Okay, so let's talk about these DNA inserts. What do we mean by DNA inserts now? We'll talk about libraries. Now, DNA libraries are basically, well, it's the same concept as a library, where, for example, you're interested in reading a book, so you check where this book is, and you find out that the book exists on the fourth floor, I'm sorry, rack number whatever, shelf number whatever. So you go there, you pick up the book that you're interested in. And the same idea exists in DNA. That is, we have plasmids that can contain, each one of them containing a different a DNA piece that is different from other plasmids. So, and we can uh, uh, save, store these plasmids in fridges, on shelves, whatever, you name it. And the idea is that if I'm interested in studying a certain piece of DNA or a specific gene, we can go to room number whatever, shelf number whatever, and we take the tube that contains this plasmid of interest. So, and the idea, and there are two different types of libraries. There are genomic libraries and there are cDNA libraries. Now, let's start with genomic DNA. Genomic DNA is basically, well, the whole DNA of a cell or a group of cells. We take this genomic DNA and it's cut by a certain restriction nuclease. We have a plasmid, we combine these cut DNA molecules with each other. Now we have each DNA piece inserted into a plasmid. We take these plasmids and we transform bacteria. And each bacterial cell would contain a recombinant DNA, a plasmid DNA, that is different from the other cell. Now, each one of them would have a different piece cloned into the plasmids that they carry. So that's genomic DNA library. Now, I would know later on what each bacterial cell or clone contains exactly, what piece of DNA they contain. Okay, and anytime I want to study this piece of DNA, all I need to do is take this bacterial cell or cells and grow them up, and I'll have millions and billions of copies of the plasmid DNA that contains the piece of DNA that I'm interested in. Well, let's talk about the cDNA libraries then. cDNA libraries are created from messenger RNA. So here we're talking about expressed genes. Expressed genes. Okay, so we take the messenger RNA from cells, we convert them to complementary DNA molecules. These are called cDNA molecules. Complementary DNA because they are complementary to the messenger RNA molecules. So we convert mRNAs to complementary DNA molecules. And we use a special enzyme known as reverse transcriptase. Reverse transcriptase. So a reverse transcriptase, is a viral enzyme. So note how we take advantage of creatures, God's creatures. So we take this viral reverse transcriptase, and this is the enzyme that converts messenger RNA into cDNA. It can convert actually any RNA molecule into DNA. So it's the reverse of transcription. Okay, and that's why we call it reverse transcriptase. So we convert messenger RNA to cDNA. Okay, and we have different genes, right? We have different messenger RNA molecules. We have different DNA molecules now, complementary DNA molecules. We add the restriction in the nuclease, creating cohesive ends. Same thing with plasmids. We combine them with each other with DNA ligase, and now we have plasmid molecules, recombinant plasmid molecules. Each one of them carries a gene. Okay. And we can transform bacterial cells with these plasmids. Now each bacterial cell now contains recombinant plasmid DNA that carries a specific gene. Okay, so we call this cDNA library. Now I know what each bacterial cell or clone carries, and every time I want to study a gene, all I need to do is pick up this bacterial cell to grow them up. Now I have millions, billions of copies of the plasmid DNA molecules of interest. Again, genomic DNA. We take genomic library. Where genomic DNA is cut by a restriction nuclease. The same endonuclease is used to cut plasmid DNA. We combine them with each other. Now we have different DNA molecules carrying different fragments of genomic DNA of different sizes, of course. Okay, and we transform these bacterial cells with the plasmids, and each clone carries a piece of genomic DNA that is different from the other cells. Now with the cDNA library, we take the messenger RNA, and this messenger RNA is converted into complementary DNA or cDNA, and we do the same thing. We cut the DNA pieces with the restriction of the nuclease. We have recombinant DNA. Each piece of plasmid molecule contains a specific gene. We transform bacterial cells again, and we have cDNA library. Okay, again, you can read this on your own. It's the same basic idea, but it's written and different figure. Same idea. That's creating a cDNA library. Again, same idea. Okay, so you can read it on your own. Now, let's talk about this point, and this point is really important. What are the differences between cDNA library and genomic library? Now, notice that with the genomic DNA, we take everything. Every DNA, we take DNA, genomic DNA. We take everything. So we can have pieces that can represent genes, exons, introns, centromeres, telomeres, promoter proximal elements, enhancers, uh, everything. Okay. Now, with the cDNA library, we're only taking the expressed genes. So if I take from a person a nerve DNA, genomic DNA, from nerve cells or from epithelial cells, skin cells, or whatever, and I cut the genomic DNA with a restriction in the nucleus, and I use it to create recombinant DNA or to create a library. Now, these libraries are going to be exactly identical or the same because they all contain the same DNA. But if I create a cDNA library from nerve cells, it's going to be quite different from a cDNA library created from skin cells because these different cells express different genes. Again, we have 20,000, let's say 20,000 protein coding genes, right? So we have 5,000 genes expressed in nerve cells. We have 6,000 genes expressed in skin cells. They're not going to be identical. Some of them are going to be the same, like histone genes, for example. Actin genes may be identical, and so on. But there are specific genes that are expressed in skin cells, and these are different from genes expressed in nerve cells. Okay, like neurotransmitters, for example, like the enzymes that create pigments in skin cells, and so on. Right? So basically, cDNA libraries are going to be different when created from different cells. Now, the other thing is cDNA libraries would contain things like exons only. They're not going to have promoters because they are not expressed. We're talking about RNA. representing messenger RNA molecules now. Okay, we're not talking about any type of RNA. So here we're talking about protein coding genes starting with their messenger RNA molecules. So they're going to be, the CDN library is going to represent exons. There will be no promoters, there will be no enhancers, no promoter proximal elements, no centromeres, no telomeres, just exons, no introns even. Okay, so keep that in mind. First, we'll be talking about the production of human proteins in bacteria. Okay, so remember the things that we talked about before in terms of cloning, how we can clone DNA into a plasmid. But we will start talking about the production of complementary DNA or cDNA. Now, in order to express a specific gene in bacteria, a human gene that is, we have to select its messenger RNA. Now, there are two problems when it comes to using the messenger RNA, a human messenger RNA, for any particular gene. The first problem is that there are so many RNA molecules inside the cell. Now, I told you before that like 50 years ago, about three RNA molecules were known, and these were messenger RNA, tRNA, and ribosomal RNA. Then other molecules, other RNA molecules, since then have been discovered. Now, so far we have like 15, 20 different types of RNA molecules, and there are so many of them in terms of number of molecules inside cells. So we have the messenger RNA, we have the classical ribosomal RNA and tRNA, but we have other types of non-coding RNA, that is RNA molecules that, again, that are transcribed but not translated. And they can be small and long. We talked about the long non-coding RNA molecule, long non-coding RNA molecule. Now, I did mention circular RNA briefly. Nothing much to talk about so far in terms of circular RNA, its nature and what it does exactly. Now, we have the small non-coding RNA molecules. We have the microRNA. Okay, we have the microRNA. We talked about the microRNA in our previous lecture. So we have all of these RNA molecules. Now, how can we select messenger RNA molecules specifically? Well, messenger RNA molecules are probably the only ones that have a poly A tail. All of the other RNA molecules do not have a poly A tail. So what we do is that in order to convert RNA to DNA, and then we can use this DNA molecule as a gene to express a human protein in bacterial cells, we use reverse transcriptase, which it's the vital enzyme that converts RNA to DNA. And this enzyme requires a primer, just like DNA polymerase does. So we use a poly-T primer. Okay, so we add this poly-T primer, and the reverse transcriptase synthesizes the first strand of DNA. And that's the solution. This is how we can select, this is how we can selectively use messenger RNA, but not all of the other RNA molecules. This is how we use the poly-T primer again for reverse transcriptase to synthesize the first DNA strand. Okay, and that's the solution. Problem number two is that we have introns in genes. So if I want to synthesize DNA from DNA, if I want to synthesize a gene starting with the genomic DNA, I'm gonna have a problem with introns. So, and this is the reason why we use mature messenger RNA, meaning that a messenger RNA that is polyadenylated. So what we do is that we don't use DNA to make copies of a gene because of the presence of introns. We do not use the primary messenger RNA, the RNA that contains introns and exons. Rather, we use mature messenger RNA um, that contains only exons connected to each other without any introns. And what we do, again, we use reverse transcriptase, that's the solution, to synthesize the DNA from the messenger RNA, and this messenger RNA is already spliced. So we make, again, double-stranded DNA, which is known as the complementary DNA. So now we have solved two problems. The problem number one is how we select messenger RNA and disregard all of the other types of RNA molecules. And the second problem is how we can get rid of introns. Well, we use mature messenger RNA that is already spliced. So now we have a complementary DNA that is basically a gene that contains only the exonic parts, only the coding parts. We take this complementary DNA and then we can clone it into a plasmid. Now, the whole purpose, again, is to synthesize human proteins inside bacteria. Remember, that's bacteria do not have introns and exons. Bacteria do not have their messenger RNA molecules do not have poly A. So, but now we have a cDNA that represents a human gene without introns and without the poly A, by the way, because we can remove it, we can get rid of it. So we clone it. So basically, what we do is that to express a human gene inside bacteria, we use an expression vector. Well, the vectors that we used before for cloning genes or for cloning DNA, these are called cloning vectors. The whole purpose of cloning vectors is just to amplify DNA or to have copies of DNA amplified inside bacterial cells. Regardless of what this DNA is, it can represent introns, exons, promoters, enhancers, you name it. But here we want to express a human protein, starting with human messenger RNA that is already spliced. So we convert the messenger RNA to cDNA, and then we clone the we clone the complementary DNA into an expression vector. Now this expression vector has the same components as cloning vectors: the origin of replication, a cloning site, and an antibiotic resistance gene that would allow us to select bacteria that contain this plasmid. But expression vectors have three additional components. Component number one is a promoter sequence, a promoter that bacteria can handle. Now remember, it doesn't have to be the same promoter as the original gene, okay? So like insulin gene, for the human insulin gene, I don't have to use the insulin gene promoter to express the insulin gene inside bacteria. I can use bacterial promoters. Molecules are stupid. I can fool them. So I'm going to have a promoter that functions, that works. And then I need a cloning site, of course, where I can place that gene downstream of the promoter. So I'm going to have the promoter right here. Downstream of the promoter, I can clone the gene. Okay. And then once I express the gene, In order to translate the messenger RNA in bacteria, we need a Shine-Dalgarno sequence. This is the ribosomal binding sequence. This is the sequence where the small ribosomal subunit binds to. Remember that? So go back to the transcription lecture. So I need this sequence, the Shine-Dalgarno sequence, the SD right here. Okay, I need that. And remember, we need a transcription termination sequence, not a stop codon. I'm not talking about that. We need a transcription termination sequence so that the RNA polymerase that synthesizes the messenger RNA knows that it has to stop mRNA synthesis right there. So we can clone the human gene right downstream of the promoter. The Shine-Dalgarno sequence would be part of the messenger RNA, but then I have an AUG start codon right here, and we're going to have a termination sequence downstream of where the gene is cloned, so that transcription is terminated. And of course, now the cDNA, well, we can have a stop codon right here as well, so that we would have a termination of translation as well. Now, question for you though. Question for you. Um, and I think I asked you this question before. Will the cDNA contain the stop codon for translation or not? Well, regardless, eventually what we're going to have is the following. So we can clone the plasmid, or we can transform bacterial cells with the plasmid. That is, we insert the plasmid into bacterial cells, and bacterial cells are going to recognize the promoter region, and they're going to start RNA synthesis or messenger RNA synthesis, transcription of the messenger RNA, and then they're gonna translate the messenger RNA into a protein. So we're gonna have the human protein expressed inside bacterial cells. Now, again, cells are stupid. We can fool them. Bacteria thinks. We use a bacterial promoter, by the way. Okay, not the insulin gene promoter, for example. So bacteria can be fooled so that they would transcribe the gene of interest, this one right here, starting with their promoter region. And they're gonna, in transcription, they're gonna translate the messenger RNA into a protein, starting with because there's a Shine-Dalgarno sequence and so on. And there's stop codon, start codon, stop codon, same thing as what bacteria is used to. And protein can be folded. So actually, this has worked really well, because now we can produce, by the way, the human insulin protein inside bacteria, growth hormone, plasminogen activator for blood clotting, and so on. So we can we convert bacterial cells into factories that can produce human proteins, and these proteins are used clinically to treat patients. If I take the bacterial cells and I open them up and I isolate, purify, extract the proteins, so I isolate the proteins away from the DNA, RNA, and other molecules, and I do SDS page, sodium dodecyl sulfate, which is the detergent that denatures proteins, polyacrylamide gel electrophoresis, we can isolate proteins based on size. Okay, so you can go back to the biochemistry lecture. So here we have all bacterial proteins, and this is taken from bacteria. These proteins are taken from bacteria that do not contain the plasmid. And here we have the bacteria that contain the vector, the expression vector. And you can see the only difference is this band right here, and this band represents the human protein. So they express huge amount of human protein. Okay, so this is expression vectors. Now the problem with expressing human proteins in bacteria is that bacterial proteins do not have disulfide bonds like human proteins. That's a problem. Bacteria do not have post-translation modification. There's no glycosylation of proteins, for example. Proteins can be misfolded, especially large proteins. So bacteria cannot handle large human proteins, but they can handle small proteins. And sometimes bacteria would degrade human proteins. They would say, what is that? I cannot recognize this protein. It looks funny. It looks awkward. It looks different, so they can degrade it. Now, as a solution, we can use yeast. Yeast cells are, single cell organisms. They are eukaryotic systems, so they do what human cells do in general. So and they grow fast. Okay, and we can insert plasmids inside them. Again, cells are stupid. They look at promoters. They don't know this is plasmid or genomic DNA. They can do exactly what we want them, what we ask them to do, or what we force them to do. We can fool them again. So we can use yeast to produce human proteins because they can handle human proteins better than bacterial proteins. So this is expression of human proteins in bacteria.
So let's say that we express a protein in a bacterial cell or yeast or even human cells. Uh, the question is, how can we, one, identify this protein? Number two, how can we purify this protein? How can we isolate this protein from all of the other proteins? Well, one mechanism, one way by which we can do that is by tagging proteins. Now, what we mean by protein tagging is that we add a tag, we add a label to proteins, just like having a label on t-shirts and shirts and whatever. And these labels, they give us information about these t-shirts or whatever, okay? And they allow us, in terms of biochemistry and molecular biology, we can use these tags to identify proteins, to detect proteins inside these cells, and they may also allow us to purify and isolate these proteins from the rest of these cellular components. So we create protein hybrids, just like we do in DNA. So, so how can we tag a protein? How can we label a protein? Well, what we do is that we do the same exact thing. We use a vector, and this vector contains a tag as part of it. Okay, so it's a small piece of DNA that encodes an amino acid sequence. And when we insert the DNA fragment of interest, the gene, we inserted right where the tag exists. Okay, so now the tag becomes part of the gene itself. Then we express the gene inside bacterial cells, and the tag is synthesized along with the protein, so it becomes part of a protein. So this is a tagged or a labeled protein. Again, this is a recombinant protein, and this allows us to identify the protein, to detect the protein, or even isolate the protein. So how can we isolate the protein that is tagged? Well, we do what is known as affinity chromatography. So it's basically you have something like a cylinder, okay, and we have these beads, kharazat, okay, resin, and you have a ligand, a small molecule attached to these beads or ligands, and we we put the beads inside this cylinder, and we pass all proteins isolated from cells through this column, okay, this cylinder. And the protein that has affinity, that is strength of interaction, the protein uh, uh, that can bind to the ligand would bind to the ligand, like in the blue protein right here. All of the other proteins would pass out. And then simply what we do is that we release the protein of interest from the column. Okay, this is known as affinity chromatography, and we'll be talking about affinity chromatography later on in this course. So here we isolate a protein, we purify it from the rest of proteins, and you can refer to this YouTube video to learn more about affinity chromatography. What we can also do is to separate protein, a certain protein, from the rest of other proteins inside cells by immunoprecipitation. Now, whenever you hear the word immuno, you should know that we're talking about antibodies, and these antibodies are really specific. Okay, so they bind to other proteins really specifically. So what we do is that a we have a bunch of proteins. We have large collection of different proteins. What we do is that we add a resin and a bead again, and this bead contains antibodies attached to the surfaces of these beads. Now, when we add these beads to the protein combination, the antibodies will bind to the protein of interest very specifically. These beads are heavy, so they precipitate, بترسبوا. Okay, they go all the way down, and we can remove all of these proteins, and we would have only the beads with the protein of interest bound to the beads. Then we can simply release the protein of interest from the antibodies, and here we have a purified protein. Now this is known as immunoprecipitation. So this antibody, for example, would bind very specifically to one protein and not all of the other proteins. So really specific binding. So that's immunoprecipitation. Now, the first two techniques allow us to isolate a protein from or to purify it from a bunch of other proteins. Now, in this third technique, we can detect a protein. We can identify a protein using gel electrophoresis, just like in proteins, just like in DNA, where we separate DNA fragments according to size, we can do the same thing with proteins. So we have a gel, a special gel. Um, we apply our sample into wells. Proteins, electricity is applied, a current is applied. Proteins migrate through the gel according to size. So they look like bands, right? So each band contains like many copies of the same protein or different proteins of the same size. Okay, so we can see how pure our sample is. We can see if our protein exists in a certain sample. And again, you can watch this YouTube video or any other video, by the way. Okay, now we can take it a step further. We can do something like southern blotting. We can do what is known as immunoblotting, just like southern blotting. We have our proteins separated through a gel according to size. We transfer these proteins to a membrane, this piece of paper, and we can then add antibodies, and these antibodies have signals attached to them, just like probes having radioactive label or fluorescent label. So these antibodies would bind to the protein that they recognize very specifically, and they give us a signal. So the band lights up, it gives a signal, and we can detect this signal again, just like southern blotting. So if the antibody, which is quite specific to the protein of interest, the band would light up, and we know that the protein exists in this sample. And again, you can watch this YouTube video. Now, there are different types of tags that we can use, and they have different purposes. Um, so, for example, I'm going to focus on the ones in red right here. So, we have what is known as a green fluorescent protein that we can attach to our protein of interest. We have an enzyme known as glutathione S transferase, or we can have a small tag composed of six amino acids known as histidine. So this is known as polyhis tag. Okay. Now, and they have different purposes. So green fluorescent protein would allow us to detect the protein using fluorescence or even antibody. The glutathione protein would help us purify a protein. There's an antibody that recognizes this enzyme right here, so we can detect our protein of interest because the protein would have the transferase, and the antibody would bind to the transferase, which is attached to all protein of interest. Okay, so we would know that the protein exists in our sample. The polyhis tag, there's an antibody that is specific to these 6-histidine amino acids. So this would allow us to purify our protein of interest using affinity chromatography or detect it using an antibody. Of course, with the transferase, we can also purify our protein of interest using its substrate, the substrate for this transferase. I'm going to show you how in a second. So here's how we can purify and detect a protein that is tagged with histidine. So we have our protein with the 6-histidine amino acids as part of the protein. And this protein is highly expressed in bacterial cells. So this is gel electrophoresis for all proteins. The first two samples are the total protein content in a cell containing our protein of interest. And you can see that this is our protein, okay? uh, Highly expressed, and you can tell from the intensity of the band. A lot of protein exists in these cells. Now, after purification, so this is before purification. After purification, we have our protein of interest right here. So how is it purified? Well, simply, we have a column, affinity chromatography column. We have beads inside it, and these beads contain nickel on the surface. And this nickel binds to the 6-histidines. And there's only one protein that contains 6 histidines, one after the other. So this protein would bind to the nickel on the beads. So everything else uh, passes through the column except with so inside the column we would have beads with nickel on the surface, and the protein with the histidines attached to these beads, and then we can release this protein later on. So again, this is a sample before. These two samples are before purification. This is after releasing the protein from the column. So simply we can clone our gene um, into a plasmid. We recombine it into a plasmid. We insert the plasmid inside bacterial cells. Bacterial cells express the protein of interest. We purify the protein using something like affinity chromatography, and then we can analyze it using gel electrophoresis. Well, we can also use glutathione S transferase for the same purpose, to isolate, to purify our protein. So it's basically the same concept. Here we have a bead right here. Okay, so we have a bead. On the surface we have glutathione, which is the substrate for this enzyme. And we have our enzyme attached to our protein of interest. That's our recombinant protein using the same exact thing. We pass all proteins through the column. Now, only our protein would bind to the glutathione. Everything else would pass through the column. And then we can simply release our protein and we have it purified.
One thing we can do is that we can create a recombinant protein. Remember, recombinant means, if I say recombinant plasmid, it means that it's a plasmid that contains a non-bacterial gene or piece of DNA or something different than what a plasmid contains. A recombinant protein is a protein that contains uh, two different proteins, for example, or different parts of proteins. So we can create a recombinant protein inside bacteria or yeast or even human cells. And what we do is that, for example, we take gene A, gene B. Now, gene A will produce protein A, gene B will produce protein B. We can put them together as one gene. So, for example, we delete the stop codon right here, and we put the genes together so that when they are translated, they're translated, when the protein is translated, when the protein is synthesized, synthesis of the protein does not stop here at the stop codon. So the synthesis of the polypeptide continues. So you have both parts, both proteins expressed, synthesized, and then you have each protein fold by itself, independently of the second protein. Now, there's a linker right here, okay? There's something that links the two proteins together. And this linker does not really affect the protein folding. So we're going to have a recombinant protein, protein A and protein B produced together. And we can do this with, we can do different combinations of proteins, by the way. And I'll give you an example.
Well, not only that, but we can also use genetic engineering to produce uh, a protein that is made of different parts of proteins. And this is due to the power of domains, the concept of domains. So what is a domain? So a domain is basically it's part of a protein. And this part, this region, folds by itself. It has its own three-dimensional structure. It is self-stabilizing. It doesn't need the rest of the protein to be stable, and it folds independently. Okay, so for example, here we have a protein, and it is composed of multiple domains. Here we have a domain, and here we have another domain, and in green we have a third domain. Now if we cut this domain out of a protein, it maintains its structure and it maintains its function as well. Okay, same thing with the other domains. So we can combine different domains from different proteins using genetic engineering. So let's say here we have gene A, and gene A produces a protein with two domains, and we have gene B, which produces a protein known as protein B, and it has also two different domains. Now, we can make a recombinant gene that produces domain of one domain from protein A and a different domain but from protein B, producing a recombinant protein that is made from that is made of different domains from different proteins. Now, this domain maintains its function and structure as well, and this domain maintains its structure and function as well. And this is how we can do this in cells, in bacterial cells or yeast cells or even human cells.
There is a protein known as green fluorescent protein. It's a protein that was isolated from jellyfish. Remember, if you see these documentaries on the Sea World, there are these jellyfish that can produce fluorescence in their legs. Now, one of these proteins is known as a green fluorescent protein, and it gives a green fluorescence. This is how it looks like. So what we do is that we can take the green fluorescent protein gene, and we can clone it upstream or downstream of another gene. So when you have synthesis, we can put it in an expression vector, and you have the production of a messenger RNA, and this messenger RNA is then translated into a protein that is large. It contains the green fluorescent protein, and it contains the protein of interest. Now, this protein right here would fluoresce because this protein, the green fluorescent protein, fluoresces as well. Okay, so let me try to play this. Okay, okay, that's jellyfish, right? Beautiful movie. You can see the fluorescent proteins moving, right? So the gene that produces these proteins have been isolated. One of them is the green fluorescent protein, and we can again clone uh, the, um, the genes together. So the green fluorescent protein would become part of the protein of interest, and each one of them folds independently of the other one. Okay, that's the advantage of domains, right? We go back to the definition of a domain. So this protein right here fluoresces. Okay, so wherever this protein goes inside the cell, I can see where it's going. We can see the cell live, by the way. So, so these are some examples of proteins that have been tagged. Tagged means that we attach another protein to them, or a piece of proteins, or whatever. This is a whole cell. These are different cells that express the green fluorescent protein, and the whole cell fluoresces. This is the actin protein with the green fluorescent protein. So you can see the actin filaments fluorescing. These are, here we have mitochondrial proteins, or a mitochondrial protein tagged with the green fluorescent protein. So the green fluorescent protein is part of the mitochondrial protein, so the whole mitochondria can fluoresce. This is the tubulin protein, the alpha tubulin protein. So you can see the microtubules right here. You can see how they are organized. We can actually have organisms like fruit flies fluorescing, or rabbits or mice fluorescing. We can see tissues, by the way. This is a whole fish. So we can also have nerve cells fluorescing by letting them express the green fluorescent protein. And we can see how these nerve cells can connect with each other. So we can understand nerve connection, for example. We can understand the morphology, the structure, the shape, the organization of tissues and organs, and even whole, organisms. Anyhow, so these are some examples, just to show you the things that we can do with the green fluorescent protein.